Good afternoon, everyone. I want to add a few perspectives from FDA. Congratulate you on wonderful meeting. Share some of thoughts that were generated here, what I heard from you all. And first, let me express the sincere appreciation for NCAT, the Critical Path Institute, and all the organizers who made this meeting possible. The collaboration between NIH, FDA, and academia and industry and platform developers is really what we witness here and what is really needed to advance this field. What strikes me most about this meeting is the momentum we've seen. So this tissue chip program, I looked it up, started in 2012, and now you see that we're no longer just talking about the potential of these technologies. We are seeing real applications, real data, real progress toward regulatory acceptance. So as you saw in my yesterday presentation, ISTAND program has been seeing remarkable growth. Numbers were like 40% of ISTAND submissions are in MPS and CIVM field. 31% is in AI, and 14%, I believe, in other AIs, and some of other tools that might be actually be combination of CIVM and AI tools. So this diversity really demonstrates the breadth of innovation happening in this space. I'm particularly encouraged that good fraction of those submissions and accepted letters of intent in ISTAND are from trace MPS overDs that represent, this meeting. So this really shows that collaborative frameworks we built is working. Maybe I'll take a moment to recap what we discussed yesterday, and just repeating that ISTAND is designed for IND agnostic regulatory tools. If you develop a NAM that you want to use to waive an animal study across multiple drug development programs, not just for one specific IND, that ISTAND qualification is the right pathway. The goal is really to establish the NAM can be broadly applied to regulatory decisions. However, if you're thinking about developing an NAM primarily for internal drug development planning to guide your company's developmental decisions and strategy, this doesn't require regulatory input and it's not appropriate for formal ISTAND qualification. That said, we're still interested in learning about these innovations through other mechanisms such as critical path innovation meetings and through our Fit for Purpose program. And of course, you can absolutely engage with us through the normal IND process with NAMs. Those conversations should happen through the standard channels, your regulatory FDA submission, and we'll be talking to your review division, not through ISTAND. So one theme that emerged repeatedly, whether it is the, in the reproductive health models or cardiac systems or neural platforms, is the critical importance of clearly defining your context of use. This isn't just regulatory jargon. A well-defined context of use tells us a number of things. What specific regulatory question are you trying to answer? What population or condition are you modeling? How will this tool fit into the drug development process? What decision will be informed by the data it generates? So the teams that have been most successful in the ISTAND submissions are those who started with the unmet need, I call it a nail, and then demonstrated the technology, the hammer, was the right tool for that specific question. I was also talking about that at FDA, we are not just reviewing submissions, we are building internal expertise with these technologies. Our work spans the full maturity spectrum from well-validated system like IPS cardiomyocytes for proarrhythmic risk assessment, where we already have multi-site validation studies, published best practice recommendations, and we have ICH guidance recognition, to emerging models like our blood-milk barrier work, where there's a critical need, unmet need, but technology is very early. So this hands-on experience is valuable to us. It helped us understand not just what these tools can do, but also what limitations are, variability, how to interpret the data they generate. This directly informs how we evaluate your submissions and provide regulatory guidance. So what I've seen at this meeting reinforces something fundamental to me. No single sector can solve these challenges alone. We do need developers who understand the biology and engineering, industry who knows the drug development questions, regulators who understand drug review and can define the qualification pathway, academia who can validate and standardize. So a conversation I've witnessed, whether it's AHL matching in immune models, standardization of protocols or data sharing, demonstrate that this community understands the value of working together. So collaboration is essential to advance this NAM space. When academic teams offer to share differentiated cells or protocols, when companies validate their AI-driven prediction systems against blinded pharma data sets, when we engage in joint projects to understand drug responses in human relevant systems, this is how we build the evidence base that supports regulatory confidence. We need to think about collaborations in creative way, right? Whether it's through formal agreements, data sharing initiatives, or simply reaching out to colleagues working on complementary problems. These partnerships accelerate progress for everyone. And again, FDA is not just a regulatory body. We are also researchers and scientists who want to collaborate where appropriate to advance the science that ultimately protects public health, and that's our mission. So as we move forward, I want to highlight a few priorities in my mind. First, data sharing. We need not just published results, but access to the underlying raw data. To build regulatory confidence, we often need to verify or replicate findings internally. So making data available accelerates that process. Second, standardization, whether it's self-sourcing, quality control criteria, or readout parameters, harmonization will be the key to scalability and broader adoption. Third, validation. We need robust, positive, and negative controls, benchmark performance criteria, and multi-site reproducibility data. The cardiac field has shown us what's possible when the community comes together around validation. Fourth, very important, education. We need to continue building understanding both within FDA about these technologies and within the development community about regulatory expectations. I admit that. I want to remind you that CDER has now established the CDER NAMS Coordinating Committee. This is a central point for all things NAM related. You can email this mailbox that I showed if you need a speaker, if you want to early, input on your technology and you're looking for opportunities to present to FDA. Please email. We're a bit overwhelmed, but we will respond. So as Dr. Ruther said in her opening remarks, this is a time to be bold. We have unprecedented support for NAMS across the federal government, the FDA Modernization Act, our NAMS roadmap, the permanent establishment of ISTAND. This signals that the time is now. Boldness must be paired with rigor. The path to regulatory acceptance requires robust science, clear context of use, and evidence that these tools provide reliable, human-relevant data that improves regulatory decision-making. I'm confident that this community is up to that challenge. The work presented here, from cardiac and neural system to reproductive health, from spaceflight research to buttocks assays, demonstrate incredible innovation and dedication to this field. So as you leave here today, I encourage you to engage early with FDA through the appropriate channels. Define your context of use clearly and specifically. Build your evidence systematically with proper controls and validation. Share your data to accelerate the field. And maybe most importantly, collaborate across sectors and discipline. The future of drug development will be built on human relevant models that better predict human responses, and you are building this future. So thank you again. Looking forward to continue this conversations.